At the bottom of the Mariana Trench, more than 6 miles down, in a spot called the Sirena Deep, there's a structure sitting in the mud. It looks a little like a fist-sized ball of trash. Sand grains, fragments of glass sponge spines, the broken shells of dead microorganisms, all glued together into something that could pass for a tiny crumbling mansion. Touch it, even gently, and it collapses into a pile of wet ash. And the glue holding this fragile little house together is the organism's own feces, some of it radioactive. Here's the part that should stop you. This isn't a colony of tiny animals working together. It's not an animal at all. It's one single cell. No brain, no muscles, no nervous system, nothing that should be capable of construction. And yet it built that. So how does one cell, with none of the equipment we'd expect a builder to need, end up architecting a home, surviving levels of radiation that would wreck human tissue, and eventually becoming something the deep ocean can't live without? Start with the size problem, because it breaks a rule you probably assume is unbreakable. Single cells are supposed to be invisible to the naked eye. This one, called Syringamina fragilissima, can grow past 20 centimeters across, roughly the size of a bowling ball. It manages this by being what's called a coenocyte. Picture an office building where every interior wall has been knocked down. Instead of millions of separate little rooms, you've got one continuous open space, and thousands of managers, thousands of nuclei floating freely through a single shared fluid, all somehow keeping the place running. And even stranger, almost none of what you're looking at is actually alive. The living tissue makes up as little as 1 to 5% of the whole structure. If you picked one up, it would feel like holding a heavy glass jar, when really, all that's inside is a single strand of cotton thread. But that's not even the strangest part of this story, because for almost 100 years, science got the whole thing wrong. In 1882, off the coast of Scotland, an oceanographer named John Murray hauled one of these up from the seafloor aboard a ship called the Triton. It fell apart in his hands. He sent the remains to his colleague Henry Bowman Brady, who described and named it the following year. But nobody could figure out what it actually was. It was built from ocean floor debris, so naturalists assumed it must be some kind of sponge. Even Ernst Haeckel, one of the most respected biologists of the era, filed it under sponges after examining Challenger expedition samples. It took until modern DNA sequencing for anyone to discover the truth. This thing wasn't a sponge, wasn't a fungus, wasn't even multicellular. It was a single-celled protist distantly related to the amoeba that had been hiding inside a shell of stolen debris the entire time. And once scientists finally knew what they were looking at, they found something in it that raised a much bigger question. In 1986, researchers David Swinbanks and Yoshihisa Shirayama were studying a burrowing species called Occultamina profunda, pulled from a trench off Japan. Inside its waste tubes, they found extreme concentrations of lead-210, a radioactive isotope. The organism was absorbing a radiation dose roughly 100 times higher than what a human experiences from natural background radiation on the surface. In a person, that kind of exposure would cause serious cellular damage over time. In this cell, no visible harm at all. Nobody has ever worked out how it protects itself. And radiation isn't the only thing it hoards. Another species, Shinkaya lindsayii, concentrates mercury at 12 times the level found in the surrounding sediment, essentially living inside a core of toxic waste by choice. Inside its own living tissue, it also stockpiles thousands of microscopic, razor-sharp crystals made of barium sulfate. Why it does this, nobody actually knows. Some scientists think the crystals work like an internal compass, helping the cell sense gravity in the pitch dark. Others think it's simply where the cell locks away toxic barium so it can't cause harm. The truth is still a guess, so by this point, you'd assume this organism is purely a hazard. Fragile, toxic, radioactive, best avoided. But in 2019, off the coast of Costa Rica, marine biologists Lisa Levin and Greg Rouse discovered something that flipped the entire picture. Operating a submersible, they opened up one of these fist-sized structures and found it packed with developing embryos, not its own. A deep-sea snailfish had used a needle-like organ to inject its eggs directly inside. The abyssal plain is almost entirely flat, muddy, and empty, with nothing to hide behind and nowhere to be safe. This toxic, radioactive, single-celled structure was the only three-dimensional shelter for miles, and a mother fish had chosen it as a nursery, oxygenated and predator-free. Scientists now call it xenophyophore. The most hazardous object in that stretch of ocean turned out to be the only thing keeping something else alive. No one has ever actually watched one of these reproduce. Bring it to the surface and it disintegrates before you can find out. So somewhere down there right now, in complete darkness, these things are quietly building, hoarding, and sheltering life, and we still don't fully understand how or why. What else is holding the abyss together down there unnoticed that we haven't found yet?